Assalamualaikum and hello to all my students. Today we are going to learn about sliding filament theory. First of all, sliding filament theory is a contraction of muscle cell across the thin filament slide past the thick filament. During contraction, the sarcomere shorten and the thin and thick filament overlap to a greater degree. So, our focus on this topic are: first, to explore the molecular structure and functional features of the thin and thick filament. Second, to understand the sequence of events in cross-bridge cycle. And last, to examine the sequence of events in multiple cross-bridge cycle.
Filament theory of how a skeletal muscle contracts involves the activities of 5 different molecules plus calcium ions. The 5 molecules include myosin, actin, tropomyosin, troponin, and ATP. In addition to these 5 molecules, calcium ions are also involved in the process of muscle contraction. Now we will explore how each of these molecules participates in the contraction of a sarcomere. Click a thick filament to examine its structure. The first molecule we will look at is the protein myosin. In skeletal muscle cells, the myosin molecules are bundled together to form the thick filaments. Click the thick filament to see an individual myosin molecule. The shape of an individual myosin molecule is similar to a golf club with 2 heads. Myosin has several important functional features. The head, or cross bridge, has the ability to move back and forth. The flexing movement of the myosin head provides the power stroke for muscle contraction. Click the head of the myosin molecule to see it move. Another feature of the myosin molecule is the hinge portion of the linear tail. This allows vertical movement so that the cross bridge can bind to actin, the thin filament. Click the tail to see this movement. Click anywhere on the myosin molecule to see both activities. The cross bridge has 2 important binding sites. One site specifically binds ATP, adenosine triphosphate, a high-energy molecule. Notice the position of the cross bridge. This is called the low-energy conformation. Click the ATP molecule to see it bind to the cross bridge. The binding of ATP transfers energy to the myosin cross bridge as ATP is hydrolyzed into ADP and inorganic phosphate. Click the ATP molecule to transfer energy to myosin. Now the cross bridge is in its high energy conformation. The second binding site on the myosin cross bridge has a strong attraction for binding to actin, as we'll see later in the cross bridge cycling animation. Now that we've examined the structure of thick filaments, let's look more closely at thin filaments. Thin filaments are composed of 3 molecules: actin, tropomyosin, and troponin. Click a thin filament to examine the arrangement of these 3 molecules. Actin is the major component of the thin filament. The actin portion of the thin filament is composed of actin subunits twisted into a double helical chain. Each actin subunit has a specific binding site to which the myosin crossbridge binds. The regulatory protein tropomyosin is also part of the thin filament. Tropomyosin entwines around the actin. In the unstimulated muscle, the position of the tropomyosin covers the binding sites on the actin subunits and prevents myosin crossbridge binding. To expose the binding sites for binding with myosin, the tropomyosin molecule must be moved aside. This is facilitated by the presence of a third molecular complex called troponin. Troponin is attached and spaced periodically along the tropomyosin strand. Troponin by itself is not able to move the tropomyosin away from the binding sites. This process requires calcium ions. After an action potential, calcium ions are released from the terminal cisternae and bind to troponin. This causes a conformational change in the tropomyosin-troponin complex, dragging the tropomyosin strands off the binding sites. Click the terminal cisternae to release calcium ions and see this effect. Before seeing how all these components work together in a complete cross-bridge cycle, click the link buttons to review any of the organic molecules or calcium ions. The 5 organic molecules